So in this particular video, we are going to look at the relationship between the visual field, which you can see, the one I've tried to put here, and the retina, how the images of the visual field are arranged on the retina. So why do we need to cover this? Because if we cover this, we get to know how to locate the parts of the retina that are affected, or the neural pathway, the parts of the neural pathway that are affected in case we have anything like a disorder. So to start with, I'll write here so that we follow along. This is the visual field up here. Then we have our retina. Of course, each eye has its own retina. So this is our left side, and this is our right side. And since we are talking about visual field having four quadrants, we have upper left, upper right, lower left, lower right quadrant, like that. So we shall also divide our retina into four. So we have four quadrants as well. So each retina will view the visual field, but how the image of the visual field will be oriented on the retina will differ. So what happens is that before I proceed, I also need to talk about the retina itself. So the retina has the lateral portion and then the medial portion. The medial portion is the one that's near your nose. So we shall call it the nasal side, and then the lateral portion, which is near the temporal bone, shall call it the temporal side. So this is the temporal and this is the nasal. This is the nasal for the right, and then it's the temporal. So how will these images appear on the retina? What you need to know is that the image formed on the retina will be inverted. To be an inverted image, meaning what's up in the visual field will be formed on the lower portion of the retina. You get? So we shall have red on the lower portion, but remember we have two quadrants up here. We have the left and right. It's the same thing. Even the quadrants will be inverted in that what's on the right will be formed on the left of the retina, and what's on the left of the visual field will be formed on the right of the retina. So let's look at the red color. The red color is on the left upper. You get? And this side we are saying it has to be down. But since it's on the left, then it will be formed on the right of our left retina like this. Then meaning our green will be on the lateral side like that. So you can easily know where the yellow will be. It's on the right lower, meaning this side it will be on the left upper. Then the blue, we have it here. As simple as that. So moving on to the right retina, we are looking at the red. So even the red will be formed, this is supposed to be red. Red will be in the in the lower right, like that. Then we have our green and the left lower, like that. And then we have our blue and the right upper, and finally our yellow on the upper left. So the main point here you need to get is that whatever is on the temporal side, let's say this is the visual field, whatever is on the right side will be formed on the left of that retina, you get? So whatever is on this side will be transferred on the left. The same thing, the right of this will be transferred to the left of this eye. Then whatever is on the left of the visual field will be transferred to the right like that, but inverted. So we can have this as the simplified version of it, separating the visual fields. This is the left side, this is the right side. So looking at each side, we can see that the nozzle field of the visual field will be viewed on the temporal retina, whereas the temporal visual field will be viewed by the nozzle retina. So continuing from here, what we can note is that whatever is formed on the nozzle side of the retina will be from the lateral visual field. As you can see that on the left retina, we have this part here, these quadrats on the left formed on the nozzle side of the retina. The same thing for the right. The lateral side of the right visual field will be formed on the nozzle side of the right retina. So how is information going to be carried from the retina? As we said earlier on, that we have the optic nerve. So the optic nerve will have two divisions in each optic nerve. We'll have a division which will carry information from the temporal side, that's the lateral side, and then from the nozzle side. So whatever carries information from the nozzle side will first go through the optic chiasm like that. Even this side, the nozzle will come to the optic chiasm, whereas whatever comes from the temporal will just proceed on that same side like that. It will not go through the optic chiasm. So we have our optic chiasm here. So that's our optic chiasm. And how will these fibers from the nozzle retina proceed? So they will cross over. You have what's from the right nozzle retina will move to the left, and then what's from the left nozzle retina, it will proceed to the right like that. So we have seen that this is the optic nerve, and whatever comes after the optic chiasm shall call it the optic tract. So this will be called the optic tracts, just like that. So these are optic tracts. This entire thing is known as optic tract, whereas the first fibers are known as the optic nerve. So where are these optic tracts heading? Optic tracts will be heading to a nucleus within the thalamus, which is known as the lateral geniculate nucleus of the thalamus. So I can draw it somewhere here. Lateral geniculate nucleus of the thalamus. That's the LGN. This is the lateral geniculate nucleus of the thalamus. Remember when we're looking at sense of taste, we say that most of the information, the sensory information, is first relayed in the thalamus before being transferred to the primary cortices. So we can see here that the thalamus comes in before we move to the primary visual cortex. It can be the easiest way to interpret it, meaning information that crosses at the optic chiasm will be from the temporal visual field. I believe you understand that. Temporal visual field, if you follow this path. Nosoretina from the temporal visual field. So proceeding from the lateral geniculate body or the lateral geniculate nucleus, we shall have what we call optic radiations. These are the ones that will transfer our sensory or visual signals now to the primary visual cortex. And where do we have the primary visual cortex? It's found around and within a structure which we call the calcarine fissure. It's a structure known as a calcarine fissure. That's where we have our primary visual cortex, and it's found within. You can see this location here. You can see that it's at the back, and that's where we have our occipital lobe. So, since these optic radiations are coming from the lateral geniculate nucleus and they are going to the calcarine fissure, they can also be called the geniculocalcarine tracts. I believe you won't forget that. So we have optic radiations from our lateral geniculate nucleus moving to the primary visual cortex through those calcarine, geniculate calcarine tracts, or call it just the optic radiations. However, we also need to take note that these optic radiations are of two types. We have what we call the Meyer's loop, and we have what we call the baum's loop. The baum's loop. So they are of two types. We have the Meyer's loop and the baum's loop. When you look at your drawings, mainly the baum's loop will be represented by broken lines. Yes, broken lines. So the easiest way you can remember this is that B goes with B. So baum's loop will have um, what will be represented by broken lines, whereas the Meyer's loop will be represented by solid lines. That's how they are represented. So what is the difference between these radiations or loops? So broken lines, which are the baum's loop, easiest way to remember this, the way I get it is that B comes before M. Meaning, to me, I take baum's loop as the one that carries information from above. So this carries information from the superior retina. And if we are to refer to our drawing here, we saw that whatever goes to the inferior portion, I mean the superior retina, will come from the inferior visual field. So don't be confused when someone says that the baum's loop carries signals from the superior retina, or when someone says it carries information from the inferior visual field. Still the same thing. It's just a mastery of the pathways which we've just looked at. Inferior visual field. So all this information will be important when we are looking at the lesions and the different parts. So it means that the Meyer's loop, for it to carry information from the inferior retina, or the superior visual field. And where are they heading? So you find that this loop, the Baum's loop, according to the textbooks, they will tell you that they will project, they will have fibers that will project into the parietal lobe, whereas the Meyer's loop, they will have fibers that will go to um, you can see that this is our temporal lobe. So Meyer's to temporal, whereas the Baum's to uh, the parietal lobe. But the whole point is that optic radiations carry information in the calcarine fissure. That's why I have the primary visual cortex. However, some of these fibers will move to these lobes separately, and we shall see the importance of the information taken to these parts. So from here, we shall look at another. So this is the primary visual cortex. So right here, that this is where we have the primary visual cortex. Primary visual cortex in the occipital lobe. However, we shall have information moving to the secondary, or call them association cortices. The secondary association cortices. So the secondary association cortices are mainly found in two areas. We have one in the parietal lobe. So we shall have fibers moving from the primary visual cortex to the parietal lobe, and in here we have a stream. Sorry, these fibers that project from the primary visual cortex to the parietal lobe are what we call the dorsal stream. Dorsal stream of fibers. So I'll draw this properly. So this is known as the dorsal stream of fibers. Dorsal stream, whereas another stream will move from the occipital lobe to the inferior temporal lobe, as you see, and the way they are projected can easily tell their name, and this will be known as the ventral or ventral stream. So each of these streams has an importance. You find that these fibers within the dorsal stream that project to the parietal lobe, they will be important for mainly navigation, that is in motion, and spatial processing of information, that is the dorsal stream. Whereas the ventral stream, for them, these are the ones that got the temporal lobe, they will be important for perception. For example, perceiving someone's face. Yeah, they will be mainly for perception. Those are the fibers to the temporal lobe. And then for the dorsal stream, it's mainly for motion and spatial processing of information. So that's it mainly for the neural pathways and the relationship between the retina and the visual field. In the next part, we are going to look at the lesions and the different levels. If you have understood this, then it will be very easy for you. So, in this particular video, we are going to look at the lesions, as we said earlier. So we are going to look at the lesions. So this one will target mainly the neural pathways, but doesn't mean that we have covered everything about the disorders. I'll briefly talk about the rest of the disorders aside from those in the neural pathway. So to start with, we have seen in the previous video that we have a medial part of your eye which is close to the nose, which we call the nozzle, represented by letter N. This is the nozzle for this left eye. This is the left side, and then this is the right side. So nozzle for the left eye, then nozzle for the right eye, and then we have the part which is near your temporal bone, this side. It's the temporal side, and also temporal side. And you saw that that will be that temporal for the visual field, and nozzle for the visual field, then nozzle for the visual field, temporal for the visual field. So we saw that information from the nozzle of the visual field will be transferred to the temporal of the retina, whereas the information from the temporal of the visual field will go to the nozzle of the retina, just like that, like that. So what do we need to observe here? We are going to look at the lesions in the different parts. For example, if we have a lesion in, let's say, okay, if we have a lesion in the optic nerve, this is what we called the optic nerve. So we can see that all information that was supposed to be transferred from this particular eye will not be transferred. So we shall have blindness in that particular eye. So what do we call that? We call that monocular blindness. Mono to mean one, then ocular to mean eye, blindness, which means blindness in one eye. So that's if we have affected our optic nerve. Then we can have another defect which affects the optic chiasm here. So when our optic chiasm is affected, we just tell that all these fibers that synapse and the optic chiasm will be affected. So where are these fibers coming from? They are coming from the nozzle retina, but we saw that the nozzle retina receives information from the temporal visual field, which means that if your optic chiasm is affected, you will not be able to see anything in the temporal field in both eyes. So we call that bitemporal because it's in both eyes. Bitemporal hemianopsia. Hemianopsia. Because it's lost in both eyes, that's why you have bitemporal hemianopsia. Then if we had a lesion in the optic tracts, if we have a lesion in the optic tracts, how do we call that? So let's look at the optic tracts. Where are they getting the information? So if we see that we see this optic tract, it has a fiber carrying information from one nozzle retina and information from the other temporal retina. So you can see that this will be called the ipsilateral fiber because it's on the same side. Then this is called the contralateral side. I mean the contralateral fiber because it's coming from the opposite eye. So if we are talking about the right optic tract being affected, right? Optic tract being affected, it means what shan't we be able to see? Let's look at this. We are seeing this is carrying information from the temporal side on the left. Then if we look at this fiber, we shall see that it's coming from the nozzle side on the left. Meaning, if my right tract is affected, actually I'll be losing vision in the left visual field of each eye. Left visual field of each eye. So what do we call that? Since it's right for left, shall call it the contralateral hemianopsia. So these are lesions in the optic tracts. The same will happen if we have a lesion in the lateral geniculate nucleus. We shall have contralateral hemianopsia, which means if someone talks about uh, left contralateral hemianopsia, I missed something here. It's supposed to be homonymous because we are losing vision in the same side. Homonymous. So if someone talks about left homonymous hemianopsia, just know it's the right. If they talk about the right homonymous hemianopsia, just know it's the left tract that's affected. So let's proceed with our lesions. So we are looking at now from the lateral geniculate body or lateral geniculate nucleus, and then we have our primary visual cortex. Or it's also called the striate cortex because it has an appearance of as if it has striations. Striated cortex. It's also called the Brodmann's area. Brodmann's area 17. Brodmann's area 17. So it can be called the primary visual cortex, or the striate cortex, or Brodmann's area 17. So we earlier on said that we have two types of optic radiations. Also, we call them the geniculocalcarine tracts because we say that they're coming from the geniculate nucleus to the calcarine fissure where we have our primary visual cortex. So we said we have the Meyer's rule, and if you remember very well, we say that this carries information from the inferior visual. It's from the inferior retina. And if we are talking about inferior retina, it's the same as saying superior visual field. Then we say we have the Meyer's, the baum's loop, which carries information from the superior retina. And saying superior retina is the same as saying inferior visual field. So, if we have a lesion in the baum's loop, what do we expect to have? We expect to have loss of vision in the inferior retina. You get it? Like whatever is seen by the superior retina will be lost. Meaning, if we can't see we don't have vision in the superior retina, that would mean that we can't see anything in the inferior visual field. So, how would we call that? If we have a lesion in the baum's loop, we would call that, I'll summarize it as CHIH. You remember we talked about contralateral when we were talking about optic tracts. So anything beyond the optic chiasm will be referred to as contralateral because it's affecting opposite side. You get it? So we would refer this as the contralateral homonymous inferior hemianopsia because we are losing vision in the inferior visual field. Then the Meyers will be contralateral homonymous superior hemianopsia. That's loss of vision in the superior visual field. So someone can also call this, you find a nickname, but it's also called a pie in the sky. That is loss of vision in the superior visual field. Like someone can't see what's in the superior visual field. It's also called pie in the sky. Whereas bumps look, where you lose vision in the inferior visual field, we call this pie on the floor. So those are lesions in the optic radiations. Then we remember we had another connection here, which were the streams. We talked about the dorsal stream, which goes to the parietal cortex, the dorsal stream, and then we talked about the ventral stream, which goes to the ventral stream, which goes to the inferior portions of the temporal. And we say that the dorsal stream carries signals responsible for motion and the spatial processing. And then we said that ventral carries information for perception. So if someone has or can't interpret information, that is perceiving it, like recognizing someone's face, we can easily tell that there is something wrong with the ventral stream moving to the secondary association cortices. Whereas if someone has something wrong with the motion, that's if their eyes can't help them, we can tell that there is something wrong with the dorsal